Okay, we're going to finish off the reproductive system and the developmental aspect of the eggs. We'll talk primarily today about the female reproductive system. Last week we talked about the male reproductive system, particularly about the process of meiosis and mitosis. Now, remember the processes in the male and female reproductive system, though similar, are different in a lot of ways. And one of these processes, as we talked about, spermatogenesis and the development of each of the sperm into a potential mate with the female egg. We will see in the female reproductive system only one egg out of the process will be produced. The process of oogenesis, where the ovum production or oogenesis begins before a woman's birth, and it accelerates during puberty, and then it comes to an end during menopause. Between the periods of time between puberty and between menopause, oogenesis occurs on a monthly basis, and that's a part of the ovarian cycle. Now, again, in that process of spermiogenesis and spermatogonia, it's very different than what we'll see in the process in the female reproductive system in oogenesis. The oogonia, or the stem cell, this is the primary developmental cell of the female, will complete mitotic divisions, and we're familiar with mitosis. This occurs before birth. Then between the 3rd and the 7th month of fetal development, the daughter cells, or the primary oocytes, will prepare to undergo this process of meiosis that we alluded to last week in the male reproductive system. And this will proceed to a certain point, but it'll only go into prophase. And this is the prophase of meiosis 1. But then the process comes to a temporary halt for a period of time until further development in the female. The primary oocytes will remain in a state of what's referred to as suspended animation until the individual reaches puberty. And then at the point of puberty, you'll see rising levels of follicle-stimulating hormone. You see this being secreted from the pituitary gland, and this will start the process of the ovarian cycle. And again, go back to that illustration, and I believe I have it in this presentation as well, the illustration where I showed you gonadotropin-releasing hormone being produced in the hypothalamus. Then you look at the anterior pituitary gland, where you'll see the production of follicle-stimulating hormone, and follow the chain of events. Each month thereafter, some of the primary oocytes are stimulated to undergo further development. Not all of the primary oocytes produced during development will survive until puberty. We alluded to this before, this process of atresia, where some of the produced oogonia will not survive. They'll actually be turned into polar bodies. And we said previously, at the time of puberty, see the numbers drop to about 400,000. The rest of the primordial follicles will basically degenerate in a process called atresia. And this is just the process of oogenesis as we see in the steps. We talk about mitosis is the initial phase of this. Then the development will go at least until the first step, or prophase, of meiosis. And then we'll take the further steps after puberty. As you see the development of the primary oocyte to a secondary oocyte and then to a mature gamete, this is at a point where the egg has fully developed, and some of the eggs will not go completely through that process again. Only about 400,000 are there at development, but that number drops significantly over a period of development. In oogenesis, a single primary oocyte is produced in the ovum. 2 or 3 of these will go into non-functional polar bodies. The nuclear event in the ovaries during meiosis is the same as what we saw in the male reproductive system. Process differs in a couple of important ways. One is the cytoplasm of the primary oocyte is unevenly distributed during the meiotic divisions. Remember we talked about equational division, reductional division. But in the female reproductive system and in the development of the eggs, the process of oogenesis, we see that the cytoplasm that is present is unevenly distributed between the eggs, and thus the ovary will release a secondary oocyte rather than a mature ovum. The cytoplasm of the primary oocyte is unevenly distributed. It produces one functional ovum, which contains most of the original cytoplasm, And then the polar bodies that are left, and it would be 2 or 3 polar bodies that are left, are basically non-functional cells that will later disintegrate. So now out of this process where we saw 4 spermatids being developed in the male reproductive system, we have only 1 ovum that's produced out of this process. The ovary will then release a secondary oocyte rather than a mature egg, and the secondary oocyte will then go into a suspended phase in the metaphase of meiosis 2. And meiosis will not be completed unless fertilization occurs. Now, in the ovarian cycle, the ovarian follicles are specialized structures in the cortex of the ovaries, where both of the oocytes growth will occur. Primary oocytes are located in the outer portion of the ovarian cortex. There are coverings of the eggs referred to as tunica albuginea, and in these clusters are where the eggs will exist, or the egg nest. Each of the primary oocytes within the egg nest will be surrounded by a single layer. This is the squamous epithelial tissue, and you'll see anywhere pretty much in the smooth muscle organs where diffusion has to take place for energy sources, and these eggs will need energy sources. You'll only see a thin layer of tissue, epithelial tissue, and this is a single layer, squamous epithelial, not stratified, but single layer of squamous epithelial around the follicle cells. So the primary oocyte follicles form this primary or primordial follicle, and after sexual maturation, different groups of these primordial follicles will then be activated. The monthly process of this activation is referred to as the ovarian cycle. The ovarian cycle is divided into phases. The follicular phase, or the pre-ovulatory phase, or the luteal phase, which is the post-ovulatory phase. Your book has some illustrations that take you through each of the steps of this process. I'm not going to ask you any test question about this. This is just kind of for your own knowledge. Those of you who go further into OB will go into a lot more detail about the ovarian cycle and the developmental process that will take place there. But during the steps of the ovarian cycle, you'll see the primary, secondary follicles will develop, then a tertiary follicle will develop, then the process of ovulation will occur, development of the corpus luteum. In the ovarian cycle, you'll see activation of primary follicles, which develop into primary follicles. You'll see primary follicles will enlarge. They'll undergo a number of divisions in this process and create several layers of follicular cells, which will basically surround the oocyte. These follicular cells will develop what's called granulosa cells, and actually the male sperm will have to break down this cellular boundary before fertilization can occur. If you remember in the anatomy of the sperm, we talked about the acromosal cap that has lytic enzymes in it that will help break down this cellular boundary that separates the oocyte from the egg. So these granulosa cells will develop around the primary oocytes. Microvilli that we've talked about before from surrounding granulosa cells will mix with the oocyte. The microvilla surrounding the layers is composed of glycoproteins. The entire region that develops with these granulosa cells is referred to as the zona pellucida. The microvilla will increase the surface area available to transfer materials through the granulosa cells, which will rapidly cause an enlargement of the primary oocytes. Then we'll see that there will be a conversion, the primordial follicle to a primary follicle. This occurs under the stimulation of follicle-stimulating hormone, again from the anterior lobe of the pituitary gland. As the granulosa cells will enlarge and multiply in the adjacent cells, the ovarian stroma, a layer of thecal cells will then surround the follicle. The thecal cells and the granulosa cells work together to produce hormone, and the primary hormone produced is called estrogen. Now, many of the follicles will develop into primary follicles, but only a few will go through this process. As the transformation begins, the wall of the follicular cells will then thicken. The granulosa cells will then begin secreting a small amount of fluid. Now, this fluid is important, follicular fluid or liquor, folliculi. This accumulates in small pockets, will gradually expand, separate into inner and outer layers of the follicle. This stage is known as the development of the secondary follicle. And although the primary oocytes will continue to grow at a steady pace, you'll see the folliculus as a whole will now enlarge and will rapidly begin to develop. And this is because the fluid will start to accumulate inside of the cells. Now, again, for testing purposes, I'm not going to ask you to go through each of these steps. The production of estrogen is an important portion of this phase. I'll go back to that real quick, where the thecal cells and the granulosa cells will work together to start producing estrogen, because estrogen is going to be important in this cycle, and certainly will be important as fertilization occurs and we have a developing fetus. The formation of the tertiary follicle. Now we had a primary, secondary, now tertiary follicle. This takes 8 to 10 days after the start of the ovarian cycle. By the 10th or 14th day of the cycle, the follicles become a tertiary follicle. This is considered to be the mature aspect of the follicle. This complex will kind of span the length of the cortex. It distorts the ovarian capsule. The oocytes will project into the opening. An opening cavity called an antrum will develop inside an expanded central area inside the follicle, but this will be surrounded by a mass of granulosa cells. Until the time of the primary oocytes has been suspended in prophase, as we alluded to earlier, you see the development of the tertiary follicle. After that point, levels of luteinizing hormone will then begin, and this will prompt the primary oocyte to complete meiosis 1. Now, instead of producing 2 secondary oocytes, the first meiotic division yields a secondary oocyte and a small non-functional polar body. Then the secondary oocyte enters into what's referred to as meiosis 2, but then again it reaches that point of suspension when it reaches metaphase, and meiosis 2 will not be completed until fertilization, as we said earlier. Now, those layers of cells that surround the oocyte, generally at day 14 through 28, the secondary oocyte attaches to these granulosa cells, and they lose their connection to the follicular wall. They basically drift within the opened aspect of the cell, or within that antrum. The granulosa cells are then associated with the secondary oocytes and a protective layer called the corona radiata. And then at ovulation, the tertiary follicle will then release a secondary oocyte. The fluid that accumulates around the corona radiata then attaches to the surface of the ovary, where it'll direct contact to the projection surrounding the entrance to the uterine tube. And if you remember, the uterine tubes have what's referred to as a ciliated epithelium. We talked about in the first part of the class, we talked about cilia, finger-like projections on the epithelial tissue. These finger-like projections help move in the respiratory tract. It helps move mucus out of the respiratory tract for us to expectorate. In the female reproductive system, we see the ciliated epithelium because of this sticky follicular fluid. We said that the cilia there are responsible to help transfer the egg, the oocyte, into the uterine tube, and it will help progress it down the fallopian tube. Ovulation marks the end of the follicular phase of the ovarian cycle. Then the start of the luteal phase will begin. The emptied follicle, once this process begins, will begin to collapse, and we'll see ruptured vessels will bleed into the antrum. The remaining granulosa cells will then invade the area of proliferation in an endocrine structure referred to as the corpus luteum. And it's called that because of the color, if that's of interest. The process basically occurs under the stimulation of luteinizing hormone, again, which is produced from the anterior pituitary gland. The cholesterol contained in the corpus luteum manufactures another hormone known as progestin, which is principally progesterone, another important hormone that we'll see in the process. The corpus luteum also secretes moderate amounts of estrogen, levels not high enough to affect ovulation, but progesterone is the principal hormone that's produced during the luteal phase. Now, the function of progesterone, I'm trying to remember if I asked a question about that. I can't remember if I answered a question about that on the exam or not. I don't think I did. Progesterone's primary function is to prepare the uterus for pregnancy, stimulating the maturation of the uterine lining. Uterine lining is a tissue structure, but under the direct control or influence of progesterone, we'll see the development of more tissue, thicker tissue layer, proliferation of blood vessels will develop there. So if there is a fertilized egg and a placental develops, it can develop in a medium that's enriched with blood vessels and nutrients to be able to keep the developing fetus, continue its development. So we'll see progesterone is a very important hormone in this process. Progesterone and estrogen levels will then fall pretty quickly unless fertilization occurs. Fibroblasts will then invade this non-functional luteum and produces a pale-like tissue. Now, the importance of recognizing the pale appearance of the tissue, or the corpus albicans, is that now you can see that as the hormone levels begin to drop, that proliferation of blood vessels, proliferation of tissue that occurs will fall off markedly. Scar tissue will form. Blood vessels will be replaced by fibroblasts, and so without circulation, the tissue will begin to disintegrate. And disintegration, or the other name for that, is called involution of the corpus luteum, which marks the end of the ovarian cycle. Now, as females age, we'll see a change in the reproductive system. Many of the primordial follicles, may have developed into primary follicles, and several of the secondary follicles have been converted into secondary follicles. But generally, only one single oocyte will be released into the pelvic cavity during ovulation. At puberty, we see that number, initially at birth and early in development, we had about 400,000. At puberty, we see the numbers drops dramatically to 200,000 primordial follicles. Then 40 years later, few, if any, of the follicles remain. Only about 500 have been ovulated in the interim, so there is a significant mortality rate for the development of the oocyte in oogenesis. Oogenesis begins during the embryonic development, primary oocyte production at birth. At the month of puberty, the ovarian cycle produces one or more secondary oocytes from the pre-existing population of primary oocytes. The number of viable and responsive primary oocytes declines remarkably at that point till the ovarian cycles at the end of the age at about 45 to 55. Few, if any, remain. Just a quick overview of the anatomy there, and as we talk about the ciliated epithelial, I think that's why I put this picture up here, was that this tissue layer here where we see the transmission of the oocytes during the process, this is a ciliated boundary. There is sticky follicular fluid here, and the cilia will help move the egg down into the uterine cavity. If the sperm has been introduced into the uterus, then fertilization will take place. And again, under the influence of the estrogen and progesterone, we'll see a proliferation of the tissue lining here inside the uterus, and then implantation can take place and the fetus will develop. The uterine cycle, or the menstrual cycle, repeated series of changes in the structure of the endometrium. The uterine cycle averages about 28 days, can range between 21 and 35 days in a healthy woman and reproductive years. 3 phases of the uterine cycle: the menses, the proliferative phase, and the secretory phase. The phases of the uterine cycle occur in response to the hormones associated with the regulation of the ovarian cycle. Menses and the proliferation phase occur during the follicular phase of the ovarian cycle. The secretory phase corresponds with the luteal phase. This is the appearance of the endometrium as I pointed to you in the previous illustration. Again, this is the area, the lining, the endothelial lining that I alluded to earlier that will proliferate during this ovarian cycle. You can see how the tissue layers, as we look at it closely microscopically, what occurs during menses is this area, the blood vessels, without the influence of estrogen and progesterone, will begin to disintegrate. This tissue level will slough. During the proliferative phase, You see the development angiogenesis, where blood vessels will develop, significant tissue layers will develop and grow under the influence of the hormone, and then as the process returns through that 28 cycle, you'll see the secretory phase and then a sloughing of tissue. So in menses, the uterine cycle begins with the onset of menses, with interval marked by the degeneration of a functional zone in the endometrium. The degeneration occurs in patches caused by the constriction of spiral arteries, which will reduce the blood flow to areas of the endothelium. Now remember that progression, that angiogenesis or development of blood vessels, occurs with the influence of the hormones. Deprived of oxygen and nutrients, the secretory glands and other tissues in the functional zone will begin to deteriorate. Eventually, we'll see weakened arterial walls, rupture, and then we'll see blood pours into the connective tissue of the functional zone. Then the blood cells and degenerating tissue will then break away and enter into the uterine lumen. They'll be lost and pass through the cervical os, or the opening of the uterus, into the vagina. Only the functional zone is affected because the deeper basilar zone is provided by blood from straight arteries that will remain unconstricted. So they are not so influenced by the hormones. So there will be a tissue layer that will remain, that will then begin to proliferate again under the influence of the hormones. The sloughing off of tissue is gradual. At each site, repairs begin almost instantaneously. Nevertheless, before menses is ended, the entire functional zone has been lost. The process of endometrial sloughing is referred to as menstruation. Generally, it lasts from 1 to 7 days. Over this period, roughly 35 to 50 cc's of blood are lost. The process is relatively painless, though the constrictive process can certainly initiate cramping and initiate an inflammatory process. That so when it says it's relatively painless, I think that probably ought to be amended by the textbook. Now, dysmenorrhea is different from normal menstruation. Now, normal menstruation, again, can be have a degree of discomfort. However, dysmenorrhea is a different process. This is painful menstruation, results from uterine inflammation. And again, there will be a small degree of inflammation even in the normal process, but dysmenorrhea, significant cramps, contractions of the uterus from conditions of the adjacent pelvic structure can cause significant discomfort, and in many instances has to be medically treated. In the proliferative phase, the basilar zone, which including the basal parts of the uterine gland, survives the menstrues intact. I should go back to so you can appreciate the functional zone. When we talk about the basilar zone and the functional zone, remember the functional zone is where we're going to see the sloughing off of tissue. Most of this basilar region and obviously the myometrium is going to remain intact during that process. In the proliferative phase, you'll see the basilar zone, including the basal parts of the uterine gland, will survive the menses intact. In days after menses, the epithelial cells of the uterine gland will multiply and spread across the endometrial surface, which will restore the integrity of the uterine epithelium. Then we'll see further growth and vascularization as a result of complete restoration in the functional zone. We'll see a reorganization period will begin. Then the endometrium will enter into the proliferative phase. Restoration occurs at the same time, the enlargement of the primary and secondary follicles in the ovary. During the proliferative phase, we see stimulation sustained by estrogen, which is secreted by the developing ovarian follicles. So there has to be coordination between the development of the follicles and this area, the basal zone. By the time ovulation occurs, the functional zone is several millimeters thick. The prominent mucus glands that extend the border of the basilar zone are noted. The endometrial glands are manufacturing a mucus-rich glycogen. The specialized mucus is essential for the survival of the fertilized egg through the earliest developmental stages as an energy source. And remember, when I said the granulosal cells in the epithelial lining that develops as a very thin layer of squamous epithelium because the diffusion of energy sources has to be able to take place for the eggs to continue to develop. The entire functional zone, highly vascularized, small arteries spiraling toward the inner surface of larger arteries in the myometrium. And then the secretory phase occurs. The endometrial glands will enlarge, accelerate the rate of secretion. The arteries that supply the uterus will elongate and then spiral through tissue into a functional zone. The activity occurs again under the combined stimulatory effect of the two hormones, progesterone and estrogen, from the corpus luteum. The secretory phase begins at the time of ovulation and will persist as long as the corpus luteum remains intact. Peaks in about 12 days. Over a couple of those days, we'll see glandular activity will begin to decline, and the uterine cycle ends as the corpus luteum stops producing the stimulatory hormones. A new cycle will then begin. The onset of menses, the degeneration of the functional zone will take place, and the secretory phase lasts about 14 days. Then as a result, you can identify the date of ovulation by counting backwards 14 days from the first day of menses. Two terms, menarche and menopause. Again, the uterine cycle begins at puberty. The first cycle is known as menarche, typically occurs between the ages of 11 and 12. Cycle will continue until menopause and terminates in the uterine cycle at about the age of 45 to 55. Over the interim, the regular appearance of uterine cycles interrupted only by circumstances such as pregnancy, obviously, but illness, certain aspects of stress can do it. Also, severe protein deficits, starvation, for example, can alter the regularity of these cycles. There is a term referred to as amenorrhea that you should know. Amenorrhea, if menarche does not occur by the age of 16, or if normal uterine cycle of an adult woman becomes interrupted for 6 months or more, the condition of amenorrhea will exist. Primary amenorrhea is a failure to initiate menses. Condition may indicate development abnormalities such as non-functional ovaries or the absence of the uterus, or endocrine and genetic disorders. Can also result from malnutrition, puberty, or delayed leptin levels as well. We also are seeing more cases of amenorrhea associated with juvenile obesity as well. That's not on the presentation here, but I don't follow those cases, but I refer those cases out. But I've had a number of people report in clinic with amenorrhea at the age of 17, 18 years of age of morbidly obese females, and I typically refer those out, but there are issues with juvenile obesity as a cause of amenorrhea. Transient secondary amenorrhea can be caused by several physical and emotional stressors in the reproductive system. Apparently in the reproductive system, somehow the process gets shut off. The factors that cause this type of amenorrhea include drastic weight loss, such as in anorexia nervosa, or severe depression and grief. Amenorrhea has also been observed in marathon runners or women who engage in training programs that require sustained high levels of exertion, and reduced body fat, reduced lipid reserves also can have a secondary amenorrhea. Hormonal activity. The activity of the female reproductive system is most certainly under hormonal control, involves interplay between secretion in both pituitary gland and the gonads. The regulatory pattern in females is much more complicated than what we saw in males. And there has to be coordination of activity between the developing follicles, because remember, they're responsible for producing estrogen. And then there also has to be coordination between the development of the uterine cycles, and also coordination between the hypothalamus producing gonadotrophin releasing hormone, the anterior pituitary gland producing luteinizing and follicle stimulating hormone. So significant coordination in the various types of hormones that are being produced in the female reproductive system. Circulating hormones control female reproductive cycles, coordinating ovarian and uterine cycles to ensure proper reproductive function. If the two cycles are not properly coordinated, infertility can result. In women who fail to ovulate or cannot conceive, even if the uterus is perfectly normal, A woman who ovulates normally but whose uterus is not normal cannot support the embryo. So anywhere in that process, from the development of the eggs to the inability of the endometrial lining to sustain an adequate functional zone, in the proliferation phase where we see the development of the functional zone, if for some reason that process doesn't occur and there's not enough development of tissue, blood vessels to be able to support a fertilized egg, then you will still have infertility as well. Any abnormality in the structure of the fallopian tubes where the eggs cannot pass down from the ovaries, any abnormality in the epithelial lining, the ciliated epithelial lining that will allow eggs to pass through, or any scarring that takes place in the anatomical structures, such as with inflammatory processes such as endometriosis, or anything that causes scarring in the fallopian tubes can affect the ability of the egg to pass down and come to fertilization. So there are a number of factors that can lead to infertility in females. Gonadotrophin releasing hormone in females. As in males, we see gonadotrophin releasing hormone from the hypothalamus regulates reproductive functions in females. The gonadotrophin pulse frequency and amplitude, which is the amount that's secreted per pulse, changes throughout the course of the ovarian cycle. Changes in gonadotrophin releasing hormone pulse are controlled by primary circulating levels of estrogen and progesterone. Estrogen increases the gonadotrophin releasing pulse frequency, and progesterone will decrease it. The pituitary gland, again, we already alluded to follicle stimulating hormone and luteinizing hormone. The endocrine cells of the anterior lobe of the pituitary gland respond as each group of endocrine cell is monitored at different frequencies. We see a group of cells that are sensitive to some of the gonadotrophin releasing hormone pulses. Frequency in other cells are insensitive to these pulses. So we consider the cells responsible for follicle stimulating hormone, luteinizing hormones. Those cells are referred to as the gonadotropes. At one pulse frequency, the gonadotropes will respond preferentially to secrete follicle stimulating hormone. Others at other frequencies, luteinizing hormone is the primary hormone that's released. Both FSH and LH production occurs in pulses that follows the rhythm of the gonadotrophin releasing hormone. Gonadotrophin releasing hormone is absent or is supplied at a constant rate without pulses. Both follicle stimulating hormone and luteinizing hormone secretion will stop. So here it's completely affected by the pulse doses and not by a continuous production of gonadotrophin releasing hormone. The follicular development begins under follicle stimulating hormone stimulation. Each month some of the primary follicles will begin to develop into primary follicles. The primordial follicles will develop into primary follicles. As the follicles enlarge, the thecal cells will start producing another hormonal substance referred to estradiol. This is a steroid hormone that's key intermediate in the synthesis of estrogen and androgens. We'll see that it's important in the production of small quantities of tissue being absorbed through the granulosa cells. We also see it's absorbed by granulosa cells and converted into estrogen. A small quantity of estrogens are also secreted by interstitial cells that are scattered throughout the ovarian tissue. Circulating estrogens are bound primarily to albumin. Albumin is a protein. Remember, proteins inside the plasma, proteins such as albumin, are transporters. They will help transport certain circulating hormones around the system. So these hormones will bind to the proteins to get through the circulatory system. So we see circulating estrogen is bound primarily to albumin, and a lesser amount is carried to the gonadal steroid binding globulin, which is another protein that's in the system as well. 3 estrogens circulating in the bloodstream. You need to know the 3 estrogen. I know that is a test question. Estradiol, estrone, and estriol. The one that's most abundant in the system with the most pronounced effect on target tissue is estradiol. It's estradiol. I think that was a test question too. Estradiol is a dominant hormone to primary ovulation. estradiol synthesis, and estradiol is converted into testosterone, which is an enzyme aromatase which converts it to estradiol. Synthesis of both estrone and estriol proceed directly from androstenedione. This is the illustration that I showed you last week or 2 weeks ago when we finished the female reproductive system, the gross anatomy of it. And so it's a nice table to be able to understand and appreciate the release of hormones, starting from the hypothalamus and the release of gonadotrophin releasing hormone, anterior lobe of the pituitary gland. We'll see production of follicle-stimulating hormone and luteinizing hormone, and you can see the effects of the follicle-stimulating hormone, and you can see the effect of the luteinizing hormone. And then each of these effects, central nervous system, bone, muscle, maintenance of female secondary sexual characteristics, accessory glands, and stimulation of endometrial growth are all important factors. And then you can also follow the arrows to see what causes the feedback process, because the regulatory steps in production in this sequence, there is a negative feedback mechanism where we'll see the secretion of both estrogen affecting the release of gonadotrophin releasing hormone in a feedback mechanism. In the reproductive cycle, and early in the follicular phase, estrogen levels are pretty low. Gonadotrophin pulse frequency is about 16 to 24 per day. And I'm not going to ask you questions about how many pulses will occur, but again, just want you to recognize that gonadotrophin releasing hormone is released in pulses. At a frequency, follicle-stimulating hormone is dominated by hormones released in the anterior pituitary gland. Estrogen released by developing follicles inhibit the luteinizing hormone secretion. Secondary follicles develop, follicle-stimulating levels decline due to a negative feedback of inhibin, which again, going back to the table before, you can see secretion of inhibin here, leading to a negative feedback process where you'll see production inside the anterior pituitary gland will be affected by this feedback mechanism. And remember now, inhibin is going to be working at the level of the anterior pituitary gland, where luteinizing hormone and follicle-stimulating hormone is produced, whereas estrogen will feed back and regulate at the level of the hypothalamus and the pulse releases of gonadotrophin. Let's see. High levels of estrogen also increase the gonadotrophin sensitivity to gonadotrophin releasing hormone. At about 14 days, estrogen levels have peaked. Gonadotropes will then maximize their sensitivity. The gonadotrophin releasing hormone pulses arriving again about every 30 minutes, which results in a massive release of luteinizing hormone from the anterior pituitary gland. And three major factors affect this surge. One is the completion of meiosis 1 by the primary oocytes, the rupture of the follicular wall, and then ovulation. Typically, ovulation will be about 34 to 38 hours after these luteinizing hormone surges, roughly about 9 hours after luteinizing hormones peak. High levels of luteinizing hormone are the trigger for ovulation. Also promotes progesterone secretion and the formation of the corpus luteum. As progesterone levels rise, the estrogen levels will fall, the gonadotrophin pulses will decline, and then we'll see the pulse rates will decrease about 1 to 4 per day. The frequency of the gonadotrophin releasing pulses stimulates luteinizing hormone more than it does the follicle-stimulating hormone secretion, and the luteinizing hormone will remain in the structure and the secretory function in the corpus luteum. Now the other hormone, progesterone, moderate amounts of estrogen are secreted by the corpus luteum. Progesterone is the main hormone during the luteal phase. The primary function is to continue preparation of the uterus for pregnancy. That's highlighted, so maybe a test question. I'll look before you leave today. If the primary function is to continue the preparation of the uterus for pregnancy by enhancing blood supply to the functional zone and stimulating the secretion of endometrial glands. Progesterone levels remain high for the next week, Unless pregnancy occurs, the corpus luteum will begin to degenerate. About 12 days after ovulation, the corpus luteum becomes non-functional. Progesterone, estrogen levels fall markedly. The blood supply to the functional zone is then restricted, and as we've said before, I'm being a little redundant now, then you'll see the endometrial tissue will begin to slough off and deteriorate. Progesterone, estrogen levels will drop. Gonadotrophin pulse frequency increases, stimulating follicle-stimulating hormone secretion, again in the anterior lobe pituitary gland, and the ovarian cycle will begin. The hormonal changes involved with the ovarian cycle in turn affect the activity of reproductive tissue and organs. At the uterus, the hormonal changes maintain the uterine cycle. Declines in progesterone and estrogen level accompany degeneration of the corpus luteum during menses. The shedding of the endometrial tissue continues for several days until rising levels of estrogen stimulate the repair and regeneration of the functional zone. This is kind of what we already went through over the menstrual cycle previously. Proliferative phase will then continue until rising levels of progesterone mark the arrival of the secretory phase, and a combination of estrogen and progesterone then cause an enlargement of endometrial glands, which will increase secretory activities. Temperature is also a factor here. The monthly hormonal fluctuation causes a physiologic change in effect of the core body temperature. So during the follicular phase, when the estrogen is the dominant hormone, the basal body temperature, or the resting body temperature, is measured upon awakening in the morning, is usually about 0.3 degrees lower than during the luteal phase when progesterone predominates. At the time of ovulation, the basal body temperature declines notably, making rising the temperature over the next day or so more noticeable. As a result, if you keep records of your body temperature, you have to be a little type A to do this. Keep records of your body temperature. Few uterine cycles, women can often determine the precise day of ovulation. Now, of course, this information is important for individuals who wish to avoid or to promote pregnancy. During fertilization, typically occurs within a day of ovulation. So the oocyte's viability and the likelihood of successful fertilization decrease markedly thereafter. There are cyclic changes in follicle-stimulating hormone, luteinizing hormone. Cyclic changes in follicle-stimulating hormone, luteinizing hormone, responsible for the maintenance of the ovarian cycle. Hormones produced by the ovarian cycle in turn regulate the uterine cycle. Inadequate hormone levels, inappropriate and inadequate responses to circulating hormone levels, or poor coordination of timing of hormone production and secondary oocytes release will reduce or eliminate the chances of pregnancy.